Hi, my name is Samantha Chilton. I did my chemistry research paper on forensic science and the insight into the different preservation in buried and entombed bodies. Forensic science is when forensic scientists analyze the collected evidence throughout a criminal investigation, including a homicide, to help identify the victim, how it happened, and who caused it. They use DNA from human fluids, hair samples, and blood that can help determine the culprit or victim. They also help in other aspects. Sometimes these cases are when victims are buried or dumped, and that is when forensic anthropologists are brought on to the investigation to determine who the victim is, if it was foul play, and who did it. A new procedure that gives insight into the different preservation in buried and entombed bodies from the article written by Italian forensic caseworks. The method can estimate the age, postmortem interval, and how old the remains are when collected. It is a very complex procedure that forensic scientists perform when examining the remains. Many things can throw off the estimation of the period or age of remains, like the environment in which the remains were found, if they were left in the sun, if the bones were picked clean by animals, and if they were buried in direct soil or a coffin of some sort. Elaborate biological factors also affect change like age, body size, and trauma postmortem or after death. The reason is no accepted method of this is published. They recently conducted a study of 408 bodies that showed that the presence of zinc coffins slows the decay of a body significantly and can preserve the remains biologically and chemically in a controlled stage for decades, which influences adipocere formation. The method represents a significant problem when implementing it for postmortem interval estimation. The definition adipocere is a byproduct of a mixture of fatty acids, FFAs, that forms under perfect conditions due to hydrolysis of triglycerides in adipose tissue, fatty tissue. This is usually formed in a later stage of postmortem decomposition. Scientists conducted another experiment using pig adipose tissue, samples that were buried in the soil directly as a control. Then one was buried in a small coffin lined in plastic and satin material. Then another sample was written in polyethylene bags, polyester clothes, and cotton clothes for 12 months to see how these materials helped with decomposition slowing down. Their results showed that the adipocere formation differed in each sample when it was analyzed using infrared spectroscope, inductively coupled plasma mass spectrometer, and gas chromatograph, mass spectrometer. Infrared spectroscope is a technology that uses a part of the spectrum, including electric magnetic radiation, to alter organic molecules, vibrational and rotational covalent bonds. The inductively coupled plasma mass spectrometer measures elements at the trace level of a biological fluid. Gas chromatography, I'm really bad at pronouncing things, and mass spectrometer are tools used to identify and measure the concentration of chemicals, food, prescription drugs, fuels, and the environment, and more. The sample wrapped in the polyethylene bag did not have an adipocere formation due to the lack of dispersing properties and the consequential liquefaction of the tissue. This is also the case with the sample wrapped in cotton. Now, polyester clothing helped the formation of adipocere because it supports the accumulation of fatty acids. The scientist who conducted the experiment suggested that these types of materials helps advance the exchange of cations and induce the saponification process, which is the process in which a hydration reaction where free hydroxide breaks the ester bond between the fatty acids and glyceride of a triglyceride, resulting in the free fatty acids and glycerol, and they both are water soluble. Lastly, the adipose tissue samples buried in the different two coffins show similar results. It shows the slow formation of adipocere in correlation with the environment of the control sample and the higher decomposition rate. These studies showed the significance in investigating the effects of different burial circumstances when considering decomposition rates. And then here's all of my wonderful references. And I think that's it. Thank you for watching my video.